Good morning, doctor.
Good morning. Good morning.
Good morning, students. We'll start the session of the day. We have with us Dr. Jyoti Lakshmi Vadaseri. She's a staff scientist 5 from National Institute of Plant Genome Research, New Delhi. She has an extensive research experiences in the field of plant metabolics, metabolomics. She has a postdoc in Cornell University, USA, and PhD from Friedrich Schiller University, Jena, Germany. She has a few awards and fellowships to her credit, like PMBO Global Investigator 20 to 2024, and head Max Planck India Partner Group in Chemical Ecology. Wilmer Pfeffer Stefan Prize, German Botanical Society, awarded for the best paper in 2010. Max Planck Society postdoctoral fellowship, DAD postdoc fellowship, and PhD with Summa cum laude, Friedrich Schiller University, Germany, fellowship for International Max Planck Research School for PhD, and a junior research fellowship from ICAR, Delhi. Her research interests are to understand the molecular mechanisms by which plants defend themselves against wide array of insect herbivores, and to use this knowledge in developing insect control strategies. She is currently focusing on mechanisms underlying rapid plant perception in insect attack and mechanism of plant microbial symbiote recognition. She has more than 32 public research publications to her credit. So with this short introduction, I invite Dr. Jyoti Lakshmi to deliver the
Hello. Yeah, thank you for the introduction. I'll start sharing the screen. So is it in full screen mode?
Yes, doctor.
Yeah. Okay, so you can I'll start the talk. If the students have any query, you can ask in the end. Okay, so the title of the talk is on metabolomics for crop improvement. I am working at the National Institute of Plant Genome Research, which is located in New Delhi, and it's an institute under the Department of Biotechnology, Government of India. So we'll see how we use these new techniques. You have heard about techniques like genomics, metabolomics, proteomics. This is an example how metabolomics is used for crop improvement in our lab and worldwide. Okay, so our lab studies insect herbivory. That is how plants defend themselves against insect pest. So as you know, there are various kinds of insect pest which attacks all part of the plant. Here you can see a leaf chewing larvae eating on the leaf. Phloem sucking aphids. There are certain insects which drink the juice from inside the insect, inside the plant. There are mesophil grazing leaf miners. There are mites. There are root feeding insects and also fruit borers. So in a nutshell, they attack all parts of the plant. And as you know that the plants cannot move and they have to defend against a lot of insects and still remain alive. So however, all plant insect interactions are not bad for the plant. What here I talked about was about conflict where they are bad. So plants are food for herbivorous arthropods. However, there are certain interactions which are also good for the plant, or it's an alliance. Yeah. So for example, there are some predatory or parasitic arthropods which protect the plants from herbivore damage. For example, this is an insect egg which is deposited on a harmful, this green thing is a harmful insect. And there are other insects which are called as parasitoids which eat this insect. So they have their eggs deposited on this harmful insect. So there can also be a good relation between plant and insect, and majority of the interaction between plants and insects are good in nature. For example, you know butterflies, they are involved in pollination. So most of the angiosperms rely upon insects for successful reproduction. So when you talk about plant-insect interaction, it's both alliance and conflict. See, our lab is working more on the conflict side of this interaction where they are harmful to the plants. Some strange reason. Okay. Now, my lab works on a specific insect called Spodoptera. It is a generalist insect, which means that it can feed on many plants. It doesn't have a host specificity, so it can feed on. This is an example where it feeds on the model plant Arabidopsis. It can feed on tomato, wide array of insects it can feed on. It's common, it's called as common cutworm. You might have seen it everywhere, and its scientific name is Spodoptera litura. We also work on the fall armyworm, which is also a generalist, but it is predominantly attacking maize in India currently. And it's a huge pest of maize. It can feed on the leaf. It can, bore into the maize plants, and it's causing a lot of crop loss in India. So anyhow, they are all Spodoptera species, and they are voracious and very fast feeders, and they feed mostly on the aerial parts or on the upper part of the leaf, and it has a wide host range. Yeah. So now, as I told you, plant-insect interaction is very is a complex interaction, and plants have to somehow recognize this insect herbivory. So this is a cartoon of a plant which is being bitten by an insect. When it is biting, it is recognizing two things. It recognizes the wounding that the insect does on the leaf, and it also recognizes the elicitors or chemical molecules inside the spit of the insect. So as it eats, it deposits spit. So this or oral secretion. So the plant can recognize both the wounding and also the elicitors in the oral secretion of the insect. Now, once it recognizes it, what we know is that it activates a hormone signaling pathway called jasmonate signaling pathway. And this in turn allows the plant to activate secondary metabolism. Yes, like chemical defenses, for example, secondary metabolites or defensive proteins. This, just keep in mind that this direct defense is what makes the plant less tasty to insect. So it might activate a secondary metabolite, and the leaf is no longer tasty. Sometimes it can activate defensive proteins, which can make the digestion of the plant material difficult inside the insect gut. So it will not feed it anymore. So these are some of the strategies of direct defense. There is also something called as indirect defense, where when the insect feeds on the plant, it sends out certain chemicals into the air, which are called volatiles. And these chemicals attract predators and parasitoids. What are predators and parasitoids? I told you before, these are insects which attack this pest. Okay, insects which attack other insects. So it calls these predators or parasitoids for help via these secondary all these volatile, and they come and eat this pest. Okay, this is an example of an ant eating or attacking another pest. So there are both direct defense, or those defenses which are occurring inside the plant, and indirect defense. It is sending out, it is calling other other partners for help to defend itself. So this is what we know about plant-insect interaction in general. Now, I talked about jasmonic signaling, and that it occurs upstream of this direct defense strategy. So it's very important for you to understand that plant hormone pathway is critical for plants in multiple, all stages of its life cycle. Forget about plant-insect interaction. In general, it's important. Many plant hormones are important for fruit ripening, for seed dormancy, germination, growth, branching, flower development, many processes, and also biotic stress. Okay, so now what are these plant hormones that we are talking about? So plants inside them have small molecules which are present in very low concentration, and they can move from one part of the plant, one part to the another, and they are extremely important to plants to defend against various kind of biotic and abiotic stress. So these are examples of these plant hormones. For example, jasmonic acid isoleucine. You might have heard about salicylic acid. This is a hormone which is induced when there is a microbial insect, microbe attacking the plant. Abscisic acid, cis or PDA. So there are many hormones which are available in the plant, which helps the plant to overcome this many stresses that it faces as it lives. Now, these hormones, for example, many of these hormones are markers for in the plant for a specific stress. So when we talk about plant defense, when it defends against a microbe or an insect, the major plant hormones activated are jasmonic acid and salicylic acid. When we talk about drought stress, the first hormone which is activated in plant is abscisic acid. Just give me a moment. Hello? Yeah. They're only GC. Yeah. So there is a problem with the earthing in the institute. So the engineering is trying to, I mean, it's not the equipment, it's the earthing. So it has to be fixed by the engineering department. It will take till the end of this month, at least. Yeah. That is what Naveen Sutta is saying. Okay. I'm behind him. I'm hoping, but they are telling that it is in the main substation, which also connects with the PGF and everything. So the equipment we will not on unless the thing is solved, because otherwise it will harm the equipment. You could try in ICGP. Ranjan will have it there. Ranjan. You ask Jiten Thakur, he has GC, or Shams will have it. They're pretty fast there. Yeah. No, I did it there. No, it will be done. Yeah, I mean, I also do proteomics, many things. I just write it as, it's something special that you want to do there. They will allow. Yeah. I'm sorry, it was an urgent call. So there are other plant hormones, for example, abscisic acid, which is a marker for drought stress. There are also, hormones called as for growth hormones, for example, cytokinins, which are involved in cell division and differentiation. There are also hormones which are involved in stem. When you talk about dormancy, germination, we always think of gibberellins, which is a growth hormones. And there is also coordination of many growth processes via auxin, which is a growth hormone. So keep in mind that there are two kinds of hormones, growth hormones and defense hormones. The defense hormones help plants in defending against various stresses, and these include jasmonic acid, salicylic acid, abscisic acid, etc. The growth hormones, for example, cytokinin, gibberellin, auxin, these are the growth hormones. Now, when we talk about plant-insect interaction, jasmonic acid biosynthesis, as I told you, is very important for chewing pests. They control 90% of all defenses against Lepidopteran insects. Now, this is the structure, the chemical structure. Why I'm showing you this is to for you to get a grasp that biology is not just, it's an interdisciplinary subject. When we talk about chemical ecology, it involves a lot of chemistry as well. And this is, for example, jasmonic acid structure. It is being sensed as a bioactive moiety, which is a conjugation of jasmonic acid with the amino acid called isoleucine. So, for example, this is, as I told you, jasmonic acid controls majority of the processes upon plant-insect interaction. And this is JA. It gets converted to bioactive JA isoleucine and then into inactive form. So this bioactive JA ile is being sensed by a COI1 receptor. which then activates the downstream genes. I'll come show you how this occurs. Why I'm describing this is because it's that important for plant response against herbivory. Under normal condition, that is when the plant is not stressed, these genes or the jasmonic acid genes have to be kept under control, because too much of plant defense is not good for the growth of plant as well. So what the plant does is that it uses this JAS proteins, which are repressors, and represses this jasmonic acid responsive genes. Now, when there is a wounding, herbivory, or any other process which is dependent on jasmonic acid, its level increases. When an insect bite on the plant, or when there is a biotrophic microbe, for example, it increases the jasmonic acid level. The jasmonic acid gets converted into this isoleucine, jasmonyl isoleucine. This chemical then, or the small molecule, then enters the nucleus and binds to this special protein called COI1. Yeah, you can see the jasmonic acid isoleucine COI1. Once it's bound, it goes and degrades this JAS protein, which is a negative regulator. Okay, once this is happening, the JA responsive genes are expressed, and it starts a signaling pathway where everything downstream happens. Now, why is JA? How is JA? How did scientists find out that this is so important? This is an example where they have knocked out the COI1-1. COI1-1 is the receptor of JA. And now if this COI, or this is the gene that I was telling here, this is the receptor which binds to the small molecule JALA. Now, if you don't have this receptor, plants can no longer defend against insects. This is wild type plant which has been fed by Spodoptera larvae. And this is a COI1-1, that is the plant doesn't have this gene COI1. So now you see it is being fed more by the insect. What does that mean? It means that if the plant doesn't have this protein, it will be completely destroyed by the insect, or that JALA sensing is important. This is what they have shown with the insect weight as well. Okay, so majority of insect responsive genes are not induced in COI1-1. Now, how do you measure all these phytohormones, metabolites? When I talk about it, how do I measure it if I have to really do it? There are two methods. One is gas chromatography, mass spectrometry. Then second is liquid chromatography, mass spectrometry. Yeah, LCMS and GCMS. So mass spectrometry. So what is mass spec? It's a technique for measuring and analyzing molecules that involves introducing energy into the target molecule to cause its ionization and disintegration. So you have a metabolite inside the plant, you are bombarding it with high energy, causing it to ionize and disintegrate. The resulting primary ions and their fragments are then analyzed, and based on their mass to charge ratio, we know a molecular fingerprint. Or if you break down a molecule with high energy, it will break down in a specific pattern. And this is what we call a molecular fingerprint. Yeah. So now what are the components of a mass spectrometer? So mass spectrometer is a very expensive equipment. It includes three parts: ionization module and ion. I told you it has to be, metabolites have to be ionized by high energy. This is what it does. Then there is a module called ion separation, where they separate these metabolites according to March M/Z values, and then you have to detect them, which is the ionization detector. So we call them ion source, mass analyzer, and detector. So these are different kinds of ionization source that we have in mass spectrometry. It's not possible for you to understand everything, but keep in mind that there are electron ionization, chemical, multiple MALDI, and ion separation can occur via, ion trap, time of flight, quadruple. These are different ion separation techniques, and detection also is by various techniques. Now imagine you injected a metabolite into the plant, and you got, you got this separation into various parts. What are you going to get there? Now this is what will happen. For example, imagine this is the structure of an unknown molecule. You don't know what it is. What you will see after passing it through mass spectrometry is this thing. Three peaks you can see. This is a separation of ions based on differences in mass to charge ratio. So fragmentation of a molecule, and this is its fingerprint. For example, this molecule fragmented into CH3 ion, some fragment into different classes of ions. So this is the base. This is the it without much ionization. Then this is after it fragmented. Now this is what we call a base peak, or the peak which is in higher abundance, and it's always fragmenting at a higher ratio. So we use this as a method to quantify this metabolite based on this M over Z value. Now with phytohormones, the problem when you go to measure it is that they are organic acid, and they can usually be analyzed using GCMS. But then for doing sample preparation in GCMS, it's very tedious. You have to extract, purify, derivatize, multiple steps are there. So our institute or our lab decided to make this much easier, and we developed a method where we can quantify all the hormones in one step, a high throughput method using LCMS. So this is what we have done. So we use LCMS, quadruple LCMS, triple quad LCMS for that. And there are two kinds of LCMS that we talk about. One is a TQ and another is a TOF. So TOF is a very high precision mass spec. It's mainly used for untargeted metabolomics. We use a triple quadruple mass spec, which is used for targeted. You know what is your target, and then this LCMS is very good for its extraction. It has high sensitivity. You can analyze it in a raw extract, that is, you grind it, plant material and methanol, and do it. High selectivity of target signal and short analysis time. The extraction procedure, methanolic extract with addition of isotopically labeled standards. Yeah. So how do we quantify these metabolites? So this is the example of a run. For example, salicylic acid. The hormone will elute at this specific time, retention time 5.79, abscisic acid 6.38. So at different time, all these hormones, we make a run condition whereby they are eluting at different time, and then we used a deuterated internal standard to further quantify it. So used on a MRM based LCMS. So this is the way that we use in general. So this is in Metabolomics facility. We use it in NIPGR. It's open for all to use. And for any of your research project, you can contact the facility. Now, once I talked about hormones so far, there are other secondary metabolites which are also present in plants. For example, phenolics, terpenoids, nitrogen-containing compounds. So this is the example. This yellow ring is an example of secondary metabolites which are present in plants. So this, after photosynthesis, you get these primary metabolites, and there are secondary metabolites like coumarins, quinones, flavonoids, terpenes, alkaloids, different kinds of secondary metabolites that help plant to overcome many stresses. Now, the secondary metabolite diversity helps plant to, overcome many of its stresses. And they are many times plant specific. For example, poplar is a big tree. It has this secondary metabolite salicinoids. Vicia has lectin, Arabidopsis mainly glucosinolate and camelexin. Cocoa has tropane alkaloids. Maize has terpenes and benzoynoids. So, there are very many cases also species specific, which helps them to, overcome many insects and pests. Now, you can ask me, if plants have so much secondary metabolites, how is it that the insect is still eating on them? There are so many insects, even though they have all these secondary metabolites that are harmful to the insect. So now the insects have also, over the year during the course of evolution, have devised strategies to overcome this toxicity. Yeah, we know that secondary metabolites are toxic. So and cause health problem to the insect. So what they do, sometimes they sequester it. What is sequestration? They compartmentalize these, secondary metabolites into specific, places and detoxify it. Sometimes they rapidly excrete it. Sometimes they rapidly degrade it by attaching different chemical moieties to it. So they have various ways also to overcome the secondary metabolite based plant defense. We will see an example here of two classes of plant secondary metabolite, the glucosinolate and benzoynoids, and how they are important for plant agriculture. So glucosinolates are secondary metabolites in the cabbage family. mustard family. Essentially, you see the mustard, they're present in all of this Brassicaceae family. And this is the model plant Arabidopsis that I talk about. This is like a mouse you have in biomedical research. In plant biology, we use this plant called Arabidopsis. It is a weed, actually. It has a very short life cycle of 3 months. You can easily put in genes in there. You can do genetic transformation easily with a short life cycle, and so it is used as a model plant. So it also belongs to this mustard family. And one of the major secondary metabolites in Brassicaceae are glucosinolates. They are amino acid derived secondary metabolites, and breakdown products are toxic to insects. So this is the glucosinolate, myrocinases, and various toxic and distasteful breakdown products. So glucosinolates, as I told you, are secondary metabolites under normal conditions. That is, when there is no insect herbivory, they are physically, they come into contact with this enzyme called myrocinases in the insect when there is a feeding. But when there is no insect feeding, myrocinases and glucosinolate are physically segregated in the intact plant. They never come together unless an insect bites. Now, when there is an insect biting, the cells are broken and they are mixed. So the glucosinolate and myrocinases come in contact with each other, and this myrosinase enzyme breaks down the glucosinolates, and various toxic and distasteful products are produced. Yeah, so which is very toxic for the insect. And this is called a mustard oil bomb. Yeah, usually this bomb is never activated because this enzyme is very separated from the metabolite. But when an insect feeding happens, it explodes like a bomb. Upon tissue disruption by herbivory, this compartmentalization is compromised, and myrocinases hydrolyze the glucose moiety of glucosinolate, forming a mixture of toxic products. So this is, if you want to look at the structure, this is a glucosinolate, this is a plant enzyme myrocinases, and they convert this glucosinolate into breakdown products, one of which is called an isothiocyanate. Now, isothiocyanate is very toxic to insects, and they react with the nucleophilic site of protein and are toxic. The release of this toxic isothiocyanate from glucosinolate is referred as mustard oil bomb. Okay. So you can imagine that plants are so clever. They produce a secondary metabolite, they keep it in different compartments, stored. When there is a herbivory, they activate this bomb. Now, how do insects counteract this glucosinolate? We have heard that plants are producing cyanide. Now, how do they overcome it? So by glycosylating the glucosinolate, they either attached sugar moiety to the glucosinolate. Then, it becomes a bigger, chemically bigger structure, and then it's not possible to detoxify it. Sometimes they desulfonate the glucosinolate. You can imagine that the insect makes alteration in the structure of the core glucosinolate and avoid its glycosylation in many cases. So this is how clever insects are as well. Now, for example, here you have seen myrocinases, glucosinolate, and different breakdown products. So I've also talked about generalist insect and specialist insect. So generalist insects are insects which can feed on many plants, okay? And specialists are one which can only feed on one kind of plants. So you can ask me, how is it that this Plutella and Pyrus rape, they are only able to feed on Brassica plants if it's full of glucosinolates? Yeah, so like you can be. So for generalists, though, they they can feed on many. But what will happen to these insects if the plants have so much secondary metabolites? So this is what is shown here. You see this dotted structure is the desulfoglucosinolate. It tells us that the Plutella, which is a specialist insect, is able to convert all the glucosinolate into this desulfo form, which cannot be converted into isothiocyanate. Pyrus rape is able to convert it all into nitrile, and it cannot be detoxified. It cannot be again made to isothiocyanate. So they are able to overcome this plant, glucosinolate toxicity, at least the specialist. The generalist, they are affected greatly by the isothiocyanate. You can see the dark bars are isothiocyanate formation. Yeah. Now, plants are also very, very clever. So this is a very nice paper, which was published in PNAS, where they have imaged the glucosinolate occurring on a plant leaf. They have used a MALDI-TOF imaging, and this is an insect biting on a leaf. They took this leaf and imaged it using MALDI, and what you see in this red color is glucosinolate accumulation. Yeah, so all the red places there is glucosinolate. That is on the edges of the, where there is mid veins on the outer part of the leaf, there's a lot of glucosinolates. So what is the insect doing? It's avoiding all these parts and only eating those parts where there is no glucosinolate. Yeah, so the plants, the insects are very adapted also to the glucosinolate distribution on the leaf. So this, so I talked to you now about previously about brassicae plants and the secondary metabolite, which is specific to it, called glucosinolate, and how plants have glucosinolate, but the insect are also, some insects can detoxify it. We'll go to the next example, which is maize and its insect pests, the fall armyworm. The fall armyworm, Spodoptera frugiperda, is one of the most important maize pests worldwide. The name armyworm refers to the sudden massive occurrence of pest insect that attacks crop fields like an entire army once their previous food supply is exhausted. It causes up to 73% yield loss, and it's a major pest in India. Okay, benzoxinoid are major defense metabolites in maize. So we are involved in a project with ICAR, the Indian Council of Agriculture Research, and with their Indian Institute of Maize Research in Ludhiana. So the fall armyworm is a major pest of maize, as I told you, and there is no sources of resistance against this insect available in India. So what in this project, it's an ICR NSF project. What we are trying to do is we are trying to screen for maize plants which are resistant to this insect pest. Okay, based on what is called the Davis score rating. We have identified 2 resistant varieties which are very good, which shows very good resistance. Now, this is the 2 resistant varieties, CML71 and E63, and this is a highly susceptible variety. Yeah, this is what you can see it in the larval weight. This is the insect feeding assay. Here the insects are feeding on this plant for 8 days, and we are taking the weight of the insect after 8 days. Yeah, so you can see in the susceptible variety BML6, it has fed greatly and it has a lot of weight it has accumulated. Yeah, this is called a box plot. Okay, these dots are showing all the points where we have taken, where the weight of the insect has been taken. Yeah, and in the resistant varieties, you can see that the insects are not growing properly and they have less weight. Okay, so hence we decided to look what is making these plants resistant and what is making this susceptible. So obviously the first thing we did was to look for phytohormones. So we looked for So we look for phytohormones in the BML6, which is a susceptible variety, and the 2 resistant variety. These are the hormone levels without any insect attack. This is the control, the A panel, and the B panel is after the insect feeding. So you can very clearly see that in the A panel, the level of hormones is constitutively high in this resistant variety. Upon insect feeding, you can see it has become very red. So there is a high induction of phytohormones in the resistant variety. In the susceptible variety, there is no induction of jasmonates, and in the other resistant variety, there is a moderate induction. So this, this shows us that hormones are very critical for this resistance pattern that we see in this maize varieties. We further quantified them. This was a 24-hour feeding. We further quantified a 3-hour, 6-hour, 24-hour feeding, different times of insect feeding. Okay, so this is in the susceptible variety. You can see in control 0 time point, and in the different time points in a susceptible variety. In the resistant varieties, these 2 are resistant. In this, the induction of hormones is a little late, but it's higher than the susceptible variety, and in this E63, it's very high. So it tells me that this is the best variety, which we can use for our further crop improvement. Yeah, this is the same that we get with jasmonic acid isoleucine. So in susceptible genotype, the ML6 has reduced levels of JA and JA isoleucine. In the resistant phenotype, there's a rapid increase of JA and JA elay upon feeding. And in other resistant variety, the levels increase, but it's delayed. Yeah. So we also see that this resistant variety DMRE63 has an increased turnover of the JALA to COH JALA, and in CML71 this turnover is decreased. So in spite of it having less hormone, less JA, since this turnover is less, they are also resistant. Now, after hormones, what leads to hormones lead to secondary metabolite biosynthesis. So now in maize, the major hormone or the major secondary metabolite is what is called as benzosinoids. For example, Dimboa and Dimboa glucosides. Okay. So what do we know about this benzosinoid pathway in maize? So in maize, like in brassicaceae species, these secondary metabolites are glucosylated. They see you have added glucose to it. They are biosynthesized by this pathway, and they are stored in a vacuole or inside the cell as glucosides. That is, they are attached with sugars and they are stable. They are not problematic in this form. Now, when there is a mechanical damage, beta glucosidases in the plant will interact with the stable glucosides and result in the release of toxic glucons. Now, these guys are toxic for the insect. This is exactly like what I told you about glucosinolate in brassica. It's stored as an non-toxic form inside the plant and far away from this plant enzyme. When there is a damage, these two come together and a toxic glucon is released. Okay. Now we know all this, it's happening. And the 4 also is able, I'll skip this because I think it will be too tough for you. Now the 4, what it does is that it attaches a sugar into this Dimboa or into this secondary metabolites, and it is one of the mechanism for them to detoxify it. Yeah. Now let us ask ourselves, now how do I know that Dimboa is important or these benzosinoids are important? How do I measure it? How do I measure its levels? So targeted metabolomics by Lcms is what we did to quantify these levels of different. Now there are many kinds of benzosinoids. So we use what is called as an MRM based quantification. We have standards for all of it, and we know how these metabolites are fragmenting. So you can give that pattern onto the LCMS machine and it will detect all of these secondary metabolites in one go. Okay. So so then we went to our resistant and susceptible variety, and we found what are the levels change of benzosinoids. So you can see in the susceptible variety, upon 24 hours of feeding, there is no change in the benzosinoid levels. Whereas in the resistant, both the varieties, the benzosinoid levels were highly increased. Yeah, so this is one of the reasons why they are resistant. So increased hormones and increased secondary metabolites. Okay. So to conclude, the benzosinoids and jasmonates are excellent markers for resistance screening in plants, for example, especially in maize, which is useful for resistance to FAW. We have developed method to quantify both the metabolites, and we have confirmed the basis of the host plant resistance, and we can use it for screening much higher germplasm. So what now we are doing is identify genes in the pathway which are altered in the genotypes. So why we want to do all that? We want to utilize host plant resistance in the integrated pest management strategies against this insect. Okay. So this is the towards the end of my talk now. So this is the metabolomics facility at NIPGR that I was talking about, where we have very high-end LCMS, GC, ICP, HPTLC, etc. And we have standardized methods for quantifying hormones, amino acids, flavonoids, primary metabolites, mineral estimation. And this is open to everybody to use. It's a pay and use facility. So please feel free to, if you have any research question you want to address, you can use it here. It's also available in many such facilities are also available in many other parts of India as well. Finally, I want to thank our institute and NIPGR. This is in Delhi, my entire lab, our collaborators, Professor Matthew, Axel, Sam, and Dr. Sujay Rakshit and JC Shekhar from IMR Ludhiana, who has been involved in the FAW project. Our lab is funded by multiple funding agencies. And with this, I want to finish my talk. So you can ask me if you have any specific questions.
Thank you, Dr. Jyoti Lakshmi. The session is open for discussion. Any questions?
Can also write to me if there is any specific question that you're too shy to ask here. You can feel free to write.
No questions? In this case, then we will
Tree that can kill caterpillar. Yeah, somebody has asked, is there any tree that can kill caterpillar or harmful to it? So multiple, tree that can kill caterpillar. Yeah, so plants have multiple secondary metabolites, which is harmful to the insects. So many trees might have different kinds of secondary metabolites. Yeah, that will be harmful to a specific class of insects. Yes, but there is no one such a, where that there is no one class of secondary metabolites that can kill all insects. They are very specific for each class of plants and for each class of insects. So these examples are what we saw. So for example, fall armyworm in maize, yeah, or their benzoxenoids is only present in maize. You will not find it in mango, or you will not find it in some other plants. So these many cases, these are enough to kill the insects. All same as you saw in glucosinolate. Yeah, so it kills certain generalist insects when it gets converted into isothiocyanate. Yeah, so that's what I was trying to explain to you.
Any other questions? So we thank Dr. Jyoti Lakshmi for joining us today.
Thank you. Thank you.
Thank you so much, ma'am, for joining us.
Thank you.
So please stay with us for us. We have our next session at 11 o'clock. Dr. Amit Asthana from Naipur, Hyderabad is joining us, and he'll be giving a lecture on microfluidic devices. Okay, just
Hi, Amar. I am here. So whenever you are ready, good morning. I cannot hear you actually. 1 minute, maybe my sound is off. Yeah, right.
Okay. Good morning, Professor. How are you?
Good, good. How are you?
Yeah, good. Okay. So, students, we'll start with the second session for the day. We have with us Professor Amit Asthana, Associate Professor and Head, Department of Medical Devices, NIPER, Hyderabad. He has a PhD in chemistry from Barkatullah University. He has also a visiting research fellowship at the University of Amsterdam, Netherlands. Curie postdoctoral fellow at the Institute Curie, France. Postdoctoral fellow, postdoctoral research associate at the University of Illinois, Urbana-Champaign. Quick hire scientist at the CSIR, AMPRI, Bhopal. As a research professor at Chungam National University, South Korea. Brian Full invited professor at Chungam National University, South Korea, senior professional officer at ANFF, University of Queensland, Brisbane, Australia, Ramlinga Swamy Fellow, CSIR, CCMB, Hyderabad, and a principal scientist from 2013 to 2019 at CCMB, Hyderabad, senior principal scientist from 2019 to 2021 at CCMB, Hyderabad, and associate professor till date at NIPER, Hyderabad. So his area of interest are early diagnosis of infections, genetic disorders and metabolic disorders, and to develop affordable solutions in tools in detecting these various healthcare systems. Okay, so he is involved in developing microfluidic paper-based analytical devices or paper microfluidic devices coupled with an instrument-free or portable device, which is coupled with ambient temperature and amplification technology while using optical and electrochemical detection system for quantification of nucleic acid. So apart from this, he is also interested in developing paper-based devices as a Raman immune sensor, non-conventional methods to fabricate microfluidic devices, biopolymeric microfluidic devices for tissue engineering and cell culture, 3D printing and 3D cell culture, and site-targeted drug delivery, vectors for drug delivery and diagnosis are using microfluidic devices. So, Professor has more than 45 research papers published in reputed journals with a H index of 13, and he has 5 patents to his credits. He has worked on 7 sponsored projects on various national and international funding agencies. With this short introduction, I invite Professor Amit Asthana to deliver the talk. The floor is
thank you very much, Amar, for introduction. So I will update you very soon about my patents. So I have now 9 patents which are filed, including in 9 different places. So just give me a minute. Something is wrong with my computer. So I will start in a minute about my presentation. Just give. Okay. So as Amar has mentioned, in the meanwhile, I am trying to set up my computer. I will tell you that my major work is related to microfluidics. Now from last few years, I am trying to work on paper-based devices, and I will cover that portion today. There are several other areas where we work, but at the moment, these are the 2 areas where we are actively involved. My group is actively involved after joining NIPER. So just let me share my presentation with you. Okay. Can you see the presentation yet or not yet?
not yet.
Okay, now?
Yes.
Yes. Okay. And the panel is not coming on your side, right? Only just presentation is there, is it? Or panel is also
panel
coming?
is also coming. Go to, you can switch to the slide.
Yeah. So now it's only
yes, slide. Yes.
Right. So I will talk to you about paper-based microfluidic devices. It's my standard presentation, which I always do, but I hope you will enjoy this, and you will be able to come up with many results. I have updated it a little bit to tell you about our recent results. So I will talk about in vitro market to start with. There is a high demand for low cost medical devices or point of care devices. So when we talk about blood analyzers, like the one shown here in this slide, I will put the pointer here. This one. This is a very expensive piece of equipment which costs somewhere around a crore rupees or so, or between US dollar 500 to 5,000. This is the smallest one, but this is also expensive. When we talk about low cost, we are talking about somewhere around US dollar 10 to 1,000, 100 rupees. But these are also expensive. Now in India, we have much more advanced systems. They are very less expensive than the one which are shown here. And WHO has given a guideline for low cost POC, for especially for remote remote areas or resource limited areas, which is called as assured, where assured stand for affordable, sensitive, specific, user friendly, rapid and robust, equipment free, which is the most important one, and deliverable to end user. So all the one which are shown here in this slide on the top, they are not equipment free. So they are not assured. They still require the equipment. At certain point, you will need equipment also. But we are in my group, we are trying to work on both the things, assured as well as assured, but with E removed from there. Okay, not this one. So any of assured device, microfluidics is the way to go. Why? Because when you are using micro devices or microfluidics is nothing but when you make micro channels, micro fabricated channels, and you are trying to control the fluid in these micro fabricated devices, here the dimension become very small. So the amount which you are taking in the solution, or in your whatever you are taking, is about few nanoliters or few picoliters. I will, some device if I have with me here, I will show you. Maybe give me a minute. So I will just I pulled some so that I can show you, so you can see some of the devices live once we will start discussions. So these devices you can make. Microfluidics is the suitable platform for making any devices. Currently, if you see any microfluidic device, the type of substrate which is used are silicon, glass, plastic, textile, and paper. So textile one is developed by one group in India, and they are actually leaders now in the field of microfluidic device in India, though their textile device is still not in the market, but they are still working on it. So that is Achiria Labs in Bangalore. They have come up with a textile based paper devices, textile based devices, and they know how to manufacture it also. But for some reason, they have not yet started commercializing it, but their plastic devices are already in the market, and paper devices is the one where we work and many other groups in India work. So whenever you are working with any such devices, that device can be broken into 4 parts. One is the area of recognition, where you have receptors and molecules which identify a particular analyte, and those bioreceptor or recognition molecule can be enzyme, antibodies, microorganism. There are very less papers on microorganism as a way to identify. Cells are there, nucleic acid, aptamers, and nanoparticles always are there. Then once you will have a reaction taking place in this recognition area, you will have central signal transducers. So the devices which I have shown you before, like these ones, all have transducers or signal transducers. So they can be electrodes, like in glucometer, they have electrodes. Thermistors are when you are measuring the temperature. Photochemical detector when you are using UV detector, or vibrational detectors when you are using Raman microscopy, piezoelectric when you are using piezoelectric sensors, and pH, and so on and so forth. So you will get any signal from this, that signal will be much less, because you are talking about very minute, analyte which you are analyzing. So you have to use signal processors. So you have amplifiers, filters, multipliers, AD to analog to digital converters, unionizers, and compressors. So these things, when you add all those things, you actually come up with a small device like this. So it is not equipment free anymore. So any POC device, when you are talking about, can be just recognition area, or can consist of signal transducers, signal processor, and readout. In my group, we only work on recognition part at the moment. We do have signal transduction and signal processing, and today we will just talk of this area, and with this, can you make a device? All these POC devices currently are made up of nitrocellulose membrane, plastics, glass, silicon, etc. But they are not affordable. Though we can say that nitrocellulose membrane are affordable, but most of the nitrocellulose membranes are sourced from outside India. So say for example, GE, PAL, and many Chinese vendors are there. The only manufacturer of nitrocellulose membrane in India is MDI, Micro Devices, which is in Ambala. From there, you can also purchase nitrocellulose membrane. What we are trying to do is completely different. We are trying to work on paper. Any paper you take around you, there are so many paper. Even this tissue roll, you can make devices with this also. So that is what we are trying. We are trying to use paper, white paper, where we can make devices, and we can make those devices with wax printing, XY plotter, laser cutter, whatever you want. Some of which I will cover in my talk. Some which I have developed in my lab also I will cover, and you are free to use them. And if you have any problem, you can always contact me. And detection, of course, you can work with colorimetry, electrochemical, Raman microscopy. So we extensively work with Raman microscopy and colorimetry and naked eye detection. So the work which I will present here will be mostly naked eye and colorimetry related. Maybe if I have some slides, I don't remember. Raman microscopy, I can talk to you. And electrochemical, we are starting now. And third thing on which my group is actively working on, bioactivation of paper. So paper is an inert material, though the chemistry of paper is very well known. So if you have to put any recognition molecule on paper, you have to use immobilization. You have to do immobilization. Either it will be enzyme, aptamers, nanobodies, antibodies, reagent, or nanoparticles. So that is one part which I will briefly cover. So why paper? Because paper is inexpensive, it is renewable, okay, and it is biodegradable because it is made up of cellulose. It comes from plant material mostly. It is compostable. So when you are talking about renewable or inexpensive, that is most important part. If you want, you can just burn it and destroy it. So there will be hardly any carbon footprint of these devices. They are portable and can be stored. So how portable they are, I will give you an example. I can keep these devices, in my pocket, like these devices. These are completely paper-based devices, completely made up of paper. I am unable to see whether you just let me check once. Okay, yeah. So these are paper devices which you can see here. There are several paper devices. They are so handy. You can easily put them in pocket and take it very well anywhere you want. Third one is that they use capillaries. So whenever you have to put a drop, it will move. That I will show you a video and you will understand how it works. Okay. Now let's go to next slide. So this is a paper towel. Paper towel you see so it's a close-up, so it looks like a fabric, but it is not a fabric, but it's a paper towel. As soon as you put a drop of any liquid on this, it will weaken all the direction. It will go in all the direction without any control. Now somehow if we can make a hydrophobic barrier. So this blue area is hydrophobic area, okay, and this white area is the hydrophilic area, the paper area. Now at the end we can immobilize the recognition molecules, let it be antibodies, aptamer, enzyme, or any reagent, okay. Now if I will put a drop of sample will move within these boundaries, it will not go out. For example, here you can see I will just stop it here. If I can stop. Okay, so here you can see that this this boundary, pink boundary, which I am showing here, is actually a wax coating or wax made surface, and the center area is where I am putting the liquid, and liquid should move within these boundaries. It should move within these boundaries, and that is what will happen. So if now I allow it to move, you can see that when I'm putting the solution is moving within these boundaries and reaching to the end of this device. Here I can have recognition. I will run it once more so that you can see it without any interruption. So I'm putting the liquid inside in the port where it has to go, and it should reach to these ends where we can recognize them. So we are giving liquid a directional flow. We are forcing the liquid to flow from this point to that point. So that directional flow can happen only when you have this kind of barriers created, design created. You can create a lot of design, which we will discuss in later half of this project, this presentation. So you can see there are, if you go and search in literature, there are various way of making the devices. It has started with lithography, then people have used XY plotter where they've used PDMS, inkjet printing, plasma treatment, wax printing is very popular one, and so on and so forth. So plotting and wax printing is the one which we use a lot in our lab. And this is again a snapshot of how many fabrication methods are there. Plotting, wherein you put a pan, I will show you how it works. Inkjet printing, wax printing, Flexo printing is also one possibility, which is available everywhere in the market. So you can buy a Flexo machine and you can make your own devices. Photolithography is an expensive method. And there is a laser cutter method, which if I have slides, I will show you. Now in my lab, we mostly work on wax printing, XY plotting, and laser cutting. And today we will just talk about wax printing and XY plotting. So these are some of the devices which we have made with wax printer. These are with XY plotter. These are with again XY plotter, wax printing, XY plotter. So what we do when I say about laser printing is we made a CAD software, and this is a laser printer. This is a wax printer. So in this wax printer, unlike the other, printer where you have laser printer where you have a toner, a toner will be there. Here we have wax cubes. So this printer, Xerox used to make before. They have stopped making it. So it's a real problem. I will give you a solution also. If wax printer is not available, what you can do. There are several ways of doing it. Okay. So don't worry if I will say that some things are not available anymore. So this was the wax printer which I was using when I was working at CCMB. Okay. So I used to make the drawing with Corel Draw, any design, print it. And then once print will come, the print will be normal print. So print will, as usual print, like this printout, whatever I have in my hand. So the print is on the top of the surface, but on the other side is this, nothing is there. So now if I will, I will heat this paper, this side. If I will put it on a hot plate, this wax will melt down and it will percolate down to the other side of the paper. Okay. So what will happen is, this I will heat it, so wax will percolate down to the other side, to the complete on the other side of the paper. Okay. And it will also percolate on the horizontal direction. It will vertical direction also and horizontal direction also, it will percolate. So whenever you are making any design, you have to remember that it will percolate on the sides also. Accordingly, you have to design your device. In our case, whenever we work with wax printer, we always remember that if I'm making a 2 mm circle, then eventually after heating, I will get 1 mm circle. So my actual device will be always 1 mm. So once my devices are ready, I can put samples, reagent, I do the reaction. And if you want to do quantification, you can either take photograph with a camera, or you can use a scanner to scan it. So this is a scanner, you scan it, then you use ImageJ to measure the intensity of this, and you can plot and make a curve. Now, if you don't have this plotter, printer, then you can always buy a XY plotter. Though this is an expensive piece of equipment, there are cheaper versions also. I will again mention that to you in my next slide. Again, you take any CAD software, you make your design and use a pen. So pen will be like this. So this is my XY plotter. This is the pen. So this pen can go in these pen holders, and there is a alignment tool also, so that you can align them properly. It should touch the surface. Pen tip should touch the surface, and then you can make the device. Okay, so we put that on this plotter, plot it, because we are using a permanent ink pen here, or the ink which we have developed in our lab. So what happen is that as soon as the pen tip will touch the surface, it will percolate down to the other side, and it will spread on the other side also. So this is the single step method. You don't have to do any heating. You draw the with the XY plotter, you using permanent ink pen, like these pens, you can easily make the devices with these pens. The pen which we use extensively for making the devices is Stadler pen, which I have shown in my slide before. So Stadler pens are the pen which you should use. Write it down. Stadler permanent ink pen work the best. So I make the devices and again do the same process, and we can analyze. Now, if you don't have a wax printer, if you don't have XY plotter, you feel that this XY plotter, which is about 10 lakh rupees, is very expensive, then what you can do is you can have Met Scientist, you can go to metscientist.com and you can buy this XY plotter, which is called as XCDraw SEA3. With this also you can make designs very effectively. It's a very less expensive printer, which will cost you 40,000 or so. So you can easily fold it and put it in your backpack and take it and make the devices anywhere, and comes with pen holders and different USB cable, so that you can connect it to your computer, a power source for powering this up, and things like that. So this is about 40,000 to 50,000 rupees, in US dollar $800, which is not that expensive. There are several ink which you can use. So in place, if you don't have if you are unable to find a Stadler pen, what you can use it, you can take a sketch pen, empty the sketch pen ink, and you fill it with any waxy material and write. So we created a wax using a wax which is there. It is very hard to see here, but you can see a white color transparent material there. This is a wax which is used many a times in microfabrication industry. This wax is used when you are doing photolithography in the both side of the wafers. You need not to understand it completely, but in some cases when you are making a micro devices or MIMS devices, you have to do etching on the both side. So when you are etching the top portion, the lower portion should be protected. So we use this wax. This wax is a high temperature wax. It melts at very high temperature, and it is not soluble in any solvent. Fortunately, I found two solvents where it is soluble. So we made the solution and we fill it in a sketch pen, and we wrote with it, and we make the devices. So this is front side, this is back side of the device. This is an empty device and the filled device. I have shown you the result of this before also. Similarly, you can have several other devices. So this is how you can print. So you can see I have filled it in a sketch pen and I'm writing with this sketch pen, and you can make thousands and thousands of devices in no time. when I started my work, I had a very old printer. So I used that printer. That is the video taken by that old printer. Okay. Once devices are there, you can do many things. Like we have done blood plasma separation to start our work. We wanted to see if we can separate blood from the plasma. What you can do is you can always have antibodies added, antibody A, B, and X, and then you can separate. But what we found is even if you use 20x PBS or EDTA solution, you can separate the plasma. This is the normal device. There is no plasma separation. But whenever you have PBS, there is a plasma separation. There is leakage also sometime when devices are not made properly, like here. But you can still get some plasma. So when you have combination of antibodies plus 20x buffer, you get more plasma. We wanted to use this method to analyze glucose. So you can see I have immobilized glucose testing reagent here, which is toluene. There is reagent, and then you put a plasma here. Plasma will go and react, and it will give you orange color. This orange color intensity you can measure, and based on that you can measure the glucose concentration. So these are some of our results with glucose concentration of reagent which is already available in the market. We use that, we got nice curve, but it is only linear in the range of up to 100 mg per dl. We have stopped that work there because we wanted to use so this is with plasma. Okay, this is with the plasma, and this is what we found. We took the plasma, we took the blood, and then from blood we separated the plasma, we put it on this drop, and you can see that we were able to, when we measure the intensity was about 94 mg per dl, correspond to 94 mg per dl. So this is how we can identify the glucose. This is just one example. If you can make your own reagent, you can make this curve even longer. Then you can make a paper device where you can estimate the glucose. Now, in IIT Chennai, they didn't have these printers. So what they've done very cleverly is they found this laser printer, which is P1606DN. And this particular printer uses a cartridge, toner cartridge, which is CE278E, which has about 10% wax, less than 10% wax. So they use this device and make their paper device. So again, they have a paper, they print it, then they heat it. After heating, the wax will percolate down to the other side, and they make the complete devices. These are some example of their devices which they have made. But this printer is also not available anymore in the market. Now, I also left CCMB, so I don't have access to the wax printer which I used to have. Now, what we have done is we have purchased another printer. This is a printer which is called as HP LaserJet Pro M305DN. This printer has a cartridge, HP 778 cartridge, which has more than 15% wax. So it is better than the laser printer which I have shown. Now we are currently optimizing the process of making the devices with this printer. It has given us a very good result, but temperature and time need to be re-optimized. So that is what we are currently doing. So you can also use this printer. If you have any problem, you can contact me. My email address was there on the top, but I can share my email address again. So you can use that. So with using this printer also, you can make device. Now you have to remember, whenever you are working with any device, so these are results from permanent ink pen, but when any sample you are going to work, you should know that your devices are not leaking. I will give you one example where we had a mistake. We learned it in a hard way. But whenever you work with any blood samples or any biological fluid, you should first check whether they are leaking for your device or not. The easiest way is you make a circle. Easy ways to make the circles or squares or line kind of thing, and see if you are filling them more than the required, whether they are leaking or not. So in this particular case, I made a 5 mm circle which can hold up to 1 microliter of a sample. And I went up to 30 times more, and still these devices were not leaking. Same way we have done with methanol. So if I fill it methanol, I can see 70% methanol does not leak even if I fill it at 30 times. Okay, that means 30 microliter will just hang, but it will not leak. But when I take 100%, then it start leaking after six times more. That means about 6 microliters. Okay, so this method can be utilized for extraction process. If you want to extract something, you can use such kind of things. So you should know what is leaking and what is not leaking. So if you want some extraction to take place, maybe you can have methanol, and then this methanol will evaporate soon, and you have extracted solution in this area, and then you can do your reaction. Okay, of course this color is of the food color which we have used to visualize our results. Okay, whenever you will use SDS, please always remember SDS or any detergent will form micelles, and those micelles, because of those micelles, solution will leak out from the devices. Okay, NaCl or high ionic concentration doesn't make much difference. Only in case of blood, where ionic concentration is very high, it will leak. And this is when you have viscous solution also, it doesn't leak. So these are all the results which we have done several. What I want to show you here is that you have to do many hit and trials before you will establish your method. So these are all the things which you should check. You should check whether your device are leaking with the biofluid which you are going to use. You should also check whether your devices are leaking with the reagent which you are using. So I will give you one example of later on. Now what we have done is we have identify a biological link also. Again, this work was completely accidental. So what I want to tell you here over here is, if you get negative results also, never ever throw them away. So one person joined me for a training purpose for 6 months. He was moving to Canada, and he was a physicist, but he was very fascinated with microfluidic devices, and he wanted to work on microfluidic devices in Canada. So he came to me and he said, can I work with you and learn paper devices? So he was doing some paper devices. I was teaching him how to immobilize stuff. So when you immobilize any antibodies, then you want to cover rest of the surface. You have immobilized antibodies, and after immobilization you don't want any non-specific bonding to take place. So rest of the surface you sort of inactivate. So for inactivating, we usually use BSA or casein or any big molecule which will go and sit on the surface. So I asked him to prepare 2% BSA solution, but he has by mistake prepared 20% BSA, and when he heated that, my devices become hydrophobic. So he said, okay, this is what happened. So initially I was a little angry, but then I realized, oh, good, I think we can have another ink. We can make a hydrophobic channel with this. So what we have done is, we took bovine serum and we heated that bovine serum at around 80 degrees. It is all available in literature also. What we found is up to 60 degrees, 70 degrees, the surfaces are hydrophilic. But as soon as we will start heating beyond 80, 75, 80 degrees, the surfaces will become hydrophobic. Why? What happens is, you have a folded protein. When you heat it, it becomes denatured. And when it becomes denatured, the hydrophobic group which were coiled inside the folded protein comes out. And when they come out, your devices will become hydrophobic. So we have used the same phenomena, and when we have filled it in the pan. So this is what happened when you're working with wax. So you have wax printer or any laser printer, you print it with that, and when you will heat it at 150 degrees or 130 or 150 degrees, it will melt down and it will percolate down and it will seal the device. So this is completely sealed device. When you will put the solution will move within these boundaries only. Okay, the one we have shown you before. Now here in this case, we are writing with a wax with BSA, and because this is BSA, it is very viscous. So it's a very viscous solution by which we write. So we use the XY plotter, and then we use the correction pin. I think you people have seen the correction ink pens some time or other, like this one. This is the correction ink pen. Here, if I open this, there is a tip. if you can see the tip, there is a spring there. So when I press this spring, it is going inside. Okay. So this, we took this out, cleaned it, and oh, my hand also got some ink. So we have removed this and fitted in a syringe here, and then we have used with XY plotter. As soon as we will touch the surface, the solution will come down, and we made the design. After making the design, immediately we heated that at 80 degrees, and the protein was denatured and aggluminated, and we got the hydrophobic devices. So we have used these devices to make this method to make devices. So at 40 degrees, of course, it was leaking. 80 degrees, it was not leaking. 120 degrees also, it was not leaking. But 80 degrees is a low temperature wherein we will not lose the integrity of our device. So we are using this. We have made a lot of devices with this, and we have used this for various other applications, which I'm not covering here. So this is the tip which I was showing you, right? This is the pan which we have sealed with the device, and we were using it. Just give me a minute. Excuse me for a minute. Parita, I'm in a meeting, in a talk. Parita, I'm in a talk. Your class is at 12:30, right? 12:30, I will take the class. I'm in a meeting. 12:30, I will take your class. Right. So this is where we have done blood typing also, and sorry, blood analysis also, and you can see we can separate blood plasma also with this device. Just give me a minute. I have a class, so I have to tell them to wait. Right. So this kind of methods also, you can look for a different material, whatever you want to use, and you can use that for various applications. So we made the devices, and is the very first work which we have done, and we are still actually optimizing it. We have optimized it. We are trying to now bring this to market with the help of one company called as Micro Life Sciences, which is situated in Chennai, and with ECIC Medical College, which is employee insurance company, corporation medical college. With them, we are actually doing many tests to see whether these devices have any faults or not. So what we have done is we have done blood typing. How we have done, I will explain you. We took the printout initially, so you can see it was a sharp print. When I heated it, the diameter reduced, and I got a 1 mm by 1 mm device with a 5 mm, sorry, about 2 mm circle. And this circle we have immobilized, we have biofunctionalized this with chitosan. Okay. And then we put antibodies. So this is antibody A, this is antibody B. So whenever you will purchase antibody for blood typing, they will come with this mark only. So if this is a blue color, that means it is antibody A. If it is yellow color, it is antibody B. And RH factor always remains as colorless. If you want to read more about this work, you can always refer to this journal, International Journal of Biological Micromolecules. In 2020, this was published in volume 163. So in this case, when we make devices, we take blood sample in EDTA tubes. We put a drop of this device on this device, where A, B, C, and control is there. Control where we have no immobilization. We put it on a device and wait for some time so that it will reach to all the ends. Then once it reaches there, we wash it with saline by putting a blotting paper underneath. So this is a blotting paper which you can see underneath. We wash it, so whatever RBCs are not coagulated will be washed away. What is happening is, if I have a blood group A, what will happen here is that if I have a blood group, then my antibodies will agglutinate here, and they will precipitate, or they will get precipitated there, and they will block the capillaries of the paper. So the capillaries of the papers are 20 micron, and the agglutinated RBCs are can make much bigger clumps. So when I am washing it, the area where there is no agglutination taking place will be washed away, and rest of the area will look like this. So these are the pictures which are taken immediately, and these are some of the pictures, some of the devices which I have used. So you can still see after a long time also there is color there, actually, like this. Okay, so these are again A blood group devices which I have. These were A negative devices. Okay, because there is only on this portion there is agglutination taking place. These are A positive. Okay, I will show you more results. So if I go further, these are my results. So we have done a lot of work with various chitosan combinations, and all work very well. These are A positive, B positive, AB positive, and this is O negative, where there is no blood agglutinate in it. There is no agglutination taking place. You will see some colors on the area where we have done washing, but that does not denote anything. It should be very dark, like the one shown here. We have done testing of these different methods, and we found that our devices are stable for about 110 days, even more, 6 months also. But the device become hydrophobic, so we are now currently working on optimizing that process. Okay, now I told you that we have done a mistake when we were working on certain devices, and we found that our devices were leaking. So we did some cholesterol testing work. So cholesterol in our body remains as a cholesterol esterase. When you convert in the presence of cholesterol esterase, it turns to cholesterol, and this cholesterol in presence of oxygen will react with cholesterol oxidase to form cholesterol and hydrogen peroxide, and this hydrogen peroxide in directly detected to measure the cholesterol in the body. So the usual method is where you use 4-aminopyrene plus phenol. In presence of peroxidase, it forms a red quinone dye, which can be detected. But when we have used this with our devices started leaking. And then we went back and saw what is there which is leaking. Is cholesterol which is causing us this problem? Then we realized that it is phenol which is leaking, and we have never tested the phenol. So that is why I'm telling you, whenever you are using any reagent, you check every reagent whether they are leaking from your device or not. Then what we have done is we have removed this with orthophenylamine. Hydrogen peroxide is a very good substrate for orthophenylamine. It is used very extensively in biological reaction. In presence of peroxidase, it will give you 2,3-diamine phenylzine, which is brownish in color. So these are all result with different cholesterol concentration. And if I I will just skip this. So these are my devices with 25 to 300 DL mg per DL concentration of cholesterol, and I can easily, if I scan this, I can easily make a very nice concentration gradient and very linear gradient. But if I see by eyes, I can only see high or low. I cannot say which is the color. So there, in this particular case, you can have a device where you can tell the cholesterol is high or low, but you cannot tell whether cholesterol is in what concentration. So we also made a cholesterol total lipid profiling device where we can do cholesterol, VLGL, VLDL, sorry, and triglyceride and HDL and control. So this is the device wherein we use scanning method. So this kind of work cannot be done using, naked eye detection system. You have to have or you have to find some way of, detecting it with naked eye also. So we are currently working on this concept, whether we can do this total cholesterol method with some other method wherein we don't have to use any detection method. When we did the correlation, you can see our correlation for at least 5 samples we have done. We have not done much samples, but we got very good correlation, 0.918, 0.996 for LDL, for triglyceride 0.93, 0.973, and for HDL it was 0.93. So it was quite impressive. So that means such devices can also be utilized for total lipid profiling using a very, very simple device, and you can reduce down the cost of the analysis, and the amount of blood needed is very less. This is another work which is very close to my heart. Still in India, the pregnancy test in cattle is done by palpation method, by this, in which case a veterinarian will put a hand inside and try to feel the fetus, or you can use ultrasound, which is not possible in India. So there is no other way of calculating, or there are some devices, lateral flow devices, where you can use blood progesterone, but always remember, blood progesterone will increase or decrease. It will depend on the metabolism of a cattle, and it will fluctuate whole day. So what we have found is, we found a metabolite in fecal sample, which is a metabolite of progesterone. So progesterone, when decomposed from many metabolites, and one of them get accumulated in fecus. So when fecal sample is when fecal is extracted from the body, it is done only once in a day. So that is the whole collection of the day, and from that we can collect the we will do the analysis. So it is very simple. That antigen or metabolite which we have found is available in the market also. So what we have done is, we have in control and positive control and test zone, we have put the standard metabolite, and we have introduced antibodies which are conjugated with HRP there. And when we wash it, the amount which is not conjugated will go out, and then we have to bring the substrate. So substrate will bind there, and you will see the blue color. Okay. Now, in the case where you have a sample, fecal sample, in fecal sample there will be free antibodies, and these antibodies will preferably react with the free antibodies. When you will wash them, they will go out. Now, if I will bring the substrate, I will get either no color or very less color. So when we have to detect pregnancy in cattle, if we are getting two colors, like in the positive control and test, that means it is a non-pregnant sample. But if we get only one color, only in positive control, that means it's a pregnant sample. So we have done a lot of work. These are our non-pregnant devices. These are pregnant devices. And we have made several iteration. So this is the final iteration which we have made, but still this is hard to go into market for various reasons. So we are currently re-optimizing this method. This is the device which is made by wax printing, and the total device, every single component of this device, is made up of paper. So this is the complete device which is in my hand. We are still evaluating this, but there are certain problems which we are trying to solve with this device. So there is a video which, but I will skip this video because we don't have enough time. Okay, so, and I will quickly go to some fun work which you can also do in your lab. So we have used these paper devices for measurement of viscosity. You all know Ostwald viscosity meter. This Ostwald viscosity meter looks like this. It's a glass apparatus wherein you put the liquid, and when liquid reaches there, from there to reach to this point, we check the time. So this is the start mark and this is the end mark. Once the solution reaches to that end mark, we will calculate. So this is the formula: viscosity of water, we already know it is 1. Viscosity of test liquid, So that means that is what you have to calculate. Density of water and density of test liquid we should know, and the time taken of water from this area to this area and time taken of runoff from this area to this area you can measure. And if you want to read more about this article, you can go to Analytical Chemica Acta 2018 and 4044 volume 86 page number. So what we have done is we have made a paper device, okay, exactly copying this concept. We put a solution there, excess solution, so that it will reach down to this area, and if there is too much excess, it can go to this void area. So we, as soon as we put the liquid, we start the machine, we measure time taken from liquid from here, this point to reach to this point, and that is what we put here. And when we did, so this is the whole setup. So this is the start point, this is the end point, this is the timer which we use. You can always have some actuations here to, and it can be connected directly to this machine for actuation. And this is how we are putting the solution and measuring the time, and this is to measure the temperature. So this is how we are keeping the solution, you can see, and I will just move it faster. So this is the start point, this is the end point. When I reach to end point, this is my end point, 1.43, 1 minute 43 seconds. Okay, so this is how we measure. So what we have done is we have measured viscosity of glycerol with the OSPOL 3 repetition, with the paper device 10 repetition. You can see how accurate they are when we draw them. When we correlate them, our correlation was 0.995% for everything. You can see very nice correlation. When we do the same thing with sucrose of different percentage, we again got very impressive 0.9996. So you can make, with this paper device, you can make viscosity meter. It can be used in the labs. It can be used for measure viscosity of various other material. It can be used for measuring viscosity of bovine syrup, whether your protein is native or not. If your protein is native, that means it is good. The viscosity will be less. If it is denatured, viscosity will be more. So this can be utilized for measuring saliva viscosity also. There are various applications which we have mentioned. So with this, I would like to end my talk and would like to thank DBT, CSIR, DSIR and DOP and NIPER for the funding. If you have any questions, I will be more than happy to answer your questions.
Thank you, Professor. The session is open for discussion. Any questions you have, students?
No questions? We still have 5 minutes time. So if you have any questions, feel free, or otherwise you can always write to me to my email address. I am putting my email address on my chat, in the chat box. Okay. Feel free to ask any questions you have. If you want to understand how the devices work, if there is anything wrong. I think somebody is trying to connect to the audio. You can type also if you have any questions. I think, Amar, if there is no question,
No,
then
it's one question.
okay. What is the advantage and disadvantage? Okay, good question. See, there is always advantage and disadvantage of all the devices. With paper devices, the biggest disadvantage is that there is not a single mass manufacture of these devices. That is why these devices have not seen light of the market. Second disadvantage is the chemistry is, though cellulose chemistry is very well known, but immobilizing various molecules is very difficult. It needs a lot of optimization. But once it is optimized, it is a very, very good method to be used. Third disadvantage which I will say is, if you want to work with very pure devices, then you have to purchase this paper from Wattman or any other big company. There is one company in Chandigarh I'm trying to find out. They can supply you similar quality of material, the paper, but in a cheaper rate. So I still don't have the contact details of these people, but I'm trying to connect them. So otherwise, there is not much disadvantage. Numerous advantages are there. You can use these devices to make the diagnostic cheaper or affordable. Cheaper is not a good word. Affordable, and many things can be done on paper. I am currently working on, my group is currently working on making LAM processes, RPA process on paper. The whole amplification can be done on paper. You can extract RNA, you can do RT process, reverse transcriptase on paper devices itself, and you can do amplification also on the paper. So that means you don't have to look for a thermocycler. You're reducing down the cost. So these devices will have a lot of, and I'm sure they will see the light of the market very soon. So they are very, very, very affordable, and you can use them to reduce down the cost of the reagent also, and the amount of reagent required is also very less. I hope, Rashmi, I was able to answer your question. Thank you. Any more questions? So I think there is no more question.
Yeah. So we thank you, Professor, for joining us today. Thank you so much.
Bye.
Bye. Thank you, students, for joining us today. We have another lecture, one lecture at 2 o'clock. Okay, so please join us at 2 o'clock, 2 to 3. We have one lecture. Okay, thank you so much.